When epithelial tissues are punctured, the cells around the wound edge assemble an actomyosin cable that constricts like a purse string to draw the wound closed. Antonio Jacinto, from the Nova University in Lisbon, Portugal, studies this process in Drosophila.
So we knew that in a simple epithelia like the one we're studying, wound healing was driven largely by the formation of the contractile actomyosin cable that forms around the leading edge of the wound. And one of the main reasons for us to do this study was try to understand how the tissue make this actomyosin cable. So what happens in the tissue before this actomyosin cable forms.
Jacinto and colleagues, led by graduate student Marco Antunes, used lasers to damage the epithelial notum of Drosophila pupae. As in other simple epithelia, wounds initially expand due to the loss of tension across the tissue. Over the next 30 minutes, an actomyosin cable forms around the wound and then constricts to close it within a few hours of wounding. But Antunes et al. Focused on the first few minutes of the wound response.
And what we were surprised to see initially was a burst of actin filaments in cells around the wound. This starts a few cells away, propagates to the next cell, and flows to the leading edge of the wound, where it ends by the formation of its actomyosin cable.
The actin filaments assembled in the apical region of the epithelial cells and recruited myosin II, suggesting that they might be contractile. Indeed, Antunes et al. Observed a wave of apical cell constriction that followed the wave of actomyosin assembly as it flowed toward the wound edge. Actomyosin contractility is regulated by the Rho family of small GTPases and their effector proteins. Antunes et al. Investigated whether two effectors of Rho1, the actin-nucleating formin diaphanous and the myosin-activating kinase ROCK, were involved in regulating the flow of actomyosin assembly and cell constriction.
When we knock down these effectors using RNAi, the actomyosin flow and waves of apical constriction was stopped. The formation of the actomyosin cable is impaired and the wound takes a lot longer to close.
Rho signaling is therefore crucial to the early stages of wound healing. In other epithelia, calcium signaling has also been implicated in the immediate wound response, so Antunes et al. Used a fluorescent biosensor to monitor changes in intracellular calcium levels.
When we make this wound, there is a clear displacement of the tissue due to the release of tension, and this stress that cells suffer very likely caused stress-activated calcium channels to open, and we see an increase in intracellular calcium in those cells around the wound. We've knocked down one of such channels, TRPM, and we see that when we knock it down, there is a reduction of the calcium influx.
Inhibiting the calcium influx impaired actomyosin flow and cable formation, indicating that calcium signaling is upstream of these wound healing processes.
We see some correlation between calcium and the formation of the actomyosin flow. However, this is not simply a linear connection, and we will have to do some modeling on this process to understand how it works.
One connection between calcium signaling and actin dynamics may be the calcium-activated actin-severing protein gelsolin. When Antunes et al. Knocked down gelsolin, actomyosin flow was severely impaired after wounding, and cable formation was greatly reduced.
So with this work, we are starting to understand what are the first events that occur when you make a wound in a simple epithelia. We can start to connect the mechanical stress that the tissue suffers with molecular events that then lead to a response of the cytoskeleton to start healing the wound. Immediately after wounding, there is a dramatic increase of the levels of intracellular calcium that lead to an actomyosin flow that starts a couple of cells away from the wound and flows to the leading edge of the wound, where it contributes to the formation of actomyosin cable that then contracts to help the wound to close.
So what do Jacinto and colleagues want to investigate next?
So now we've started to do some modeling to try to understand this relationship between the calcium and the actomyosin flow in our system. And we have, in the last few years, performed the screens that affect wound healing. And now we're looking at some of those mutants to see how they can help us to understand how this process is regulated.
In the meantime, you can learn more about how epithelial wounding induces coordinated waves of actomyosin flow and apical cell constriction in the paper by Antunes et al., published in the July 22, 2013 issue of the Journal of Cell Biology.